Good morning, students. Today we will start the topic electrochemistry. Electrochemistry is a branch of chemistry which deals with the conversion of energies, namely making electrical energy to chemical energy or chemical energy to electrical energy. In electrochemistry, we come across electrolysis process, and this electrolysis process opened up many applications based on the electrolysis process. For example, we have it finds applications in electroplating, electroforming, and electroproduction of metals. So these electrochemistry is a study of basic fundamental aspects about electrochemical cells. This finds very interesting topic for scientists and also the metallurgist. Now we have to see what is electrochemical cell. So let us see the definition of electrochemical cell. Electrochemical cell is a device which converts chemical energy into electrical energy or electrical energy into chemical energy. Now based on this, there are two types of electrochemical cells. First one, galvanic cell, and second one, electrodynamic cell. Now what is this galvanic cell? Galvanic cell is a cell which converts chemical energy into electrical energy. Examples we have voltaic cell or the DNA cell. And second one, what we have is electrolytic cell. Electrolytic cell is a cell which converts electrical energy into chemical energy. Examples of battery, lead acid battery, example can be given where the electrical energy can be converted back to chemical energy. Now let us study about the galvanic cells. The galvanic cells, already we have discussed about the definition of the galvanic cells. Let us know about the construction and working of galvanic cells. Now, to construct the galvanic cell, we should take two containers. So, as you can see in the diagram, one container we have taken, and in this container we have to take zinc sulfate solution. And one more container we have to take, and in this container we have to take copper sulfate solution. So both the containers, in both the containers we have kept the rod. That is, here in zinc sulfate solution we have kept zinc anode. So zinc rod we have kept it, and that is going to act as an anode. And in copper sulfate solution we have kept copper rod, and that is going to act as a copper cathode. That is cathode. So these two, now the two solutions which are there in the containers, as you can see here, these are the two containers and these are the two solutions, that is zinc sulfate and copper sulfate. These two solutions are connected through a salt bridge. As you can see here, salt bridge. So what is a salt bridge? Salt bridge is a U-shaped tube. Salt bridge is a U-shaped tube in which we have taken saturated solution of ammonium nitrate or saturated solution of KCl, jelly-like material you can take it. And salt bridge, you can see the openings here, these openings here, both the openings can be plugged with the porous materials like cotton. So these two are so that it will be available for the movement of ions. Now these two rods, that is zinc rod and the copper rod, these are connected through the ammeter or the voltmeter through the wire. So as you can see, these two rods are connected with the help of the wire with the voltmeter. So now we have constructed the galvanic cell. Now let us know about how does galvanic cell work. Now working, we have to concentrate on the working now. Now when this is when we have constructed the galvanic cell, what will happen next is how does it work? Now zinc slowly starts dissolving and it goes into the solution as zinc 2 plus ions by liberating the solution by as zinc 2 plus ions by liberating 2 electrons. Now these 2 electrons goes to the other side of the container where copper electrode is there, and also there are Cu2 plus ions and SO4 2 minus ions. And here what will happen, these 2 ions are consumed by Cu2 plus here. Immediately it will grab and it will be converted into Cu, and that will get deposited here. So these are the reactions taking place here. What happens is zinc, okay, starts here, and there will be movement of electrons from zinc anode to copper anode. There will be movement of electrons from zinc anode towards the copper cathode. Zinc converts into zinc 2 plus 2e minus, that is by liberating electrons, and that reaction we call it as an oxidation reaction. And the other side, that is at the cathodic side, Cu2 plus ions are there. They will gain those electrons, consume those electrons, and they will convert it into Cu. They will be converting into Cu. Now these two, this is called reduction reaction. Now when we combine these two oxidation and reduction reactions, we will get the cell reaction, overall cell reaction, that is zinc plus Cu2 plus will get zinc 2 plus ions plus Cu. So this is the overall reaction, this is the electrochemical reaction. So at zinc anode oxidation reaction takes place, and at copper cathode reduction reaction takes place, and overall this electrochemical reaction takes place. So this electrochemical reaction takes place, and this is responsible for the production of electrical energy and which can be used for any external work. So here we will understand, that is, we have to keep in mind that these two are the important reactions. Those are oxidation and reduction. Oxidation, as you know, is nothing but the loss of electrons, and reduction is nothing but the gain of electrons. And here, the electrode where oxidation takes place will get negative charge. So we can say that anode is electrically negative, and here electrically negatively charged, and at the cathode reduction reaction takes place and it accepts the electrons, so that's why it is positively charged. So in galvanic cell, anode is negatively charged and cathode is positively charged. So this is how the galvanic cell works. Students, now we will understand what is meant by reference electrodes. Reference electrodes. Reference electrodes are the electrodes whose electrode potential values will be known and are used to calculate the electrode potential of other electrodes. For example, whatever you write notes or the study material what you are going to get it. So one reference book will be there. That is, we will go to the library. No? So that is one reference book is there, and from that reference book you are going to note down your matters and all that. Right? Like that, same exactly. For that reference electrodes. Those are called as reference electrodes. Reference electrodes are the electrodes whose electrode potential values will be known and are used to find out the electrode potential of other electrodes. In reference electrodes, we have two types, that is primary reference electrodes and secondary reference electrodes. Primary reference electrode. Primary reference electrodes are the electrodes again whose electrode potential values will be known to us, and for example, for this one is SHE, that is we write SHE, the full form of that one is already you might be knowing, standard hydrogen electrode. Standard hydrogen electrode, primary reference electrode. But there is a difficulty with the standard hydrogen electrode because we cannot construct the standard hydrogen electrode in the laboratories by keeping at 1 molar concentration, that becomes very difficult for us. So we, instead of primary reference electrodes, we go for secondary reference electrodes. Secondary reference electrodes. Now, secondary reference electrodes are the electrodes whose electrode potential values are going to remain constant, and are used to find out the electrode potential values of other electrodes. Now, the secondary reference electrodes we have to go because it is easy to construct here. It is easy to construct, and the potential value is reproducible here. That's why we prefer secondary reference electrodes. Secondary reference electrodes there are two types again. These are the second reference electrodes, what we call as calomel electrode is one, and we also have similar chloride electrode. But now we will, according to our syllabus, we have to concentrate more on calomel electrode. Now this calomel electrode we have to study again about the calomel electrode, the construction, working, how does it work, and what are the applications of the calomel electrode. Now let us know about the construction first. Calomel electrode. Now this is the calomel electrode, as you can see in the diagram. There are two side tubes are there in the calomel electrode. It's a long tube-like structure, and down there is a mercury. Mercury is placed at the bottom of the tube, and above that is a layer of mercury and a mercurous chloride. Above that is KCl solution. Saturated KCl solution is placed. So there is a in center we have platinum wire for external electrical contact. In the center we have platinum wire for external electrical contact, and we have side tubes, as you can see here. Side tubes are there here, and through this side tube, this KCl solution is introduced into the calomel electrode. Now, depending on the concentration of the KCl solution, we have different calomel electrode. That is, if we use a saturated KCl, then it is saturated calomel electrode. If we use 0.1 normal solution of KCl, then it is 0.1 N saturated, 0.1 N calomel electrode. Like that, because here the concentration of the, whatever the concentration of the KCl solution we take, it is designated like that: normal, decinormal, saturated. So we have decinormal, right? And if you take one N, then normal, right? So depending on that, we have this and calomel electrodes. So the construction we have to explain it like that. This calomel electrode is U-shaped tube, and we have two side tubes in that, and down there is a mercury is placed at the bottom of the tube, and above that is a layer of paste of mercury and mercurous chloride, and above that is a solution, that is a saturated solution of KCl. And center, as you can see here, there is a platinum wire for external electrical contact. There are side tubes, and through that side tubes, this KCl solution can be introduced. So this is the construction of the calomel electrode. Now we have to understand how the calomel electrode works. Calomel electrode. So here the calomel electrode is represented like this: Hg, Hg to Cl2, Cl minus. Now how does it work? Now it, depending on the nature of the other electrode which is connected, it can act as anode also, and it can act as cathode also. Now when it acts as an anode, this is the reaction. So when it acts as an anode, you can see that there is a liberation of electrons. And when it acts as a cathode, so this is the reaction, that is gaining of electrons. So these are the two reactions: when it acts as an anode and when it acts as a cathode. So the combination of this is the reversible reaction. Now the electrode potential of for calomel electrode is given by E is equals to E naught 0.0591 log of Cl minus 298 in any of the experiment, and in especially this carbon electrode is used as a reference electrode in the potentiometric titration, and along with the glass electrode it is used in the pH determination. So these are the applications of carbon electrode. Students, next we will also know about the what are concentration cells, or also called as electrolyte concentration cells. A concentration cell is a device which is made up of two electrodes, and both electrodes are made up of same element and are in contact with the same electrolyte but of different concentration. So such type of cells are called as concentration cell. I'll repeat, a concentration cell is a cell in which both the electrode, because you know that students, cell means two electrodes have to be there. So concentration cell where two electrodes, both the electrodes are made up of same element and are in contact with the same electrolyte but of different concentration. Means only the concentration varies. So such type of cells are called as, that's why the name here, as you can see, electrolyte concentration cells. Now let us take an example of copper electrolyte cell, concentration cell. Copper electrolyte, the copper sulfate, where we have taken copper sulfate. So let us have the diagram in case of this concentration cell, taking the example of copper. Now let us take the copper electrode. So let us construct the concentration cell here. So let us take one container here, and in this container we have taken copper sulfate solution, and so the concentration we have taken, that the concentration of that solution is M1. And let us take one more container. In that container also we have taken copper sulfate solution, and let us take the concentration of that solution as M2. Now these two solutions we have picked again. So both the electrodes, as I mentioned in the this definition, this concentration cell is a cell which is made up of both two electrodes, in which both the electrodes are of same metal. So here you can see that the electrode what we have dipped in this copper sulfate solution is copper, right? So which is going to act as a anode, and in one more container we have taken again copper sulfate solution, and we have taken again copper, okay, which is going to act as a cathode. So here also copper, here also copper. So this copper will act as an anode, and this copper will act as a cathode. Now this copper, both the coppers are dipped in the electrolyte, and here also we have taken copper sulfate solution, and here also we have taken copper sulfate solution. So electrodes are same, it is made up of same element, and copper sulfate also same electrolyte, but there is a difference here. So what is the difference here? The concentrations of the solution and copper sulfate solution are different. They are not the same. So that is, one of the concentration of the copper sulfate solution is M1 what we have taken, and another is M2. So different concentrations are there. So this is called as concentration cell. So this is an example for the concentration cell. The concentration cell is going to be, we can give the notation like this, is written like this: copper, a single vertical line. So it is an interface between the solution and the rod, solid rod, and where the concentration M1 we have taken, and this is solid bridge, indication to vertical line indicates that presence of a solid bridge, and C2 plus 2, and there is one more solution. Okay? That is the concentration of that solution is M2, and there is a cathode. So anode, and anode in the solution in which the anode is placed, salt bridge, solution in which the cathode is placed, and cathode. So where M1 and M2 are the molar concentrations of the Cu plus 2 ions, okay, in the half cell. Half cell because if you entirely, if you take it, it is a cell. But if you consider only 1 plus 1, it is called as a half cell. Okay. Then we can write the half cell reactions. The half cell reactions are for this concentration cells concentration cell can be written as Cu, that is copper. So what happens is it will give rise to Cu2 plus, okay, ions by liberating 2 electrons. So that is the anodic reaction, as you can see here. This is the anodic reaction. So oxidation reaction takes place at anode. And Cu2 plus, that is will accept those 2 electrons and will be reduced to copper. So that is the cathodic reaction. Reduction reaction takes place. Now these are the and oxidation takes place. Cathode reduction reaction takes place. Now when we combine these 2 reactions, that as you can see here, Cu2 plus N2, reversible reaction, Cu2 plus M1. So concentrations. So the cell reaction is a change in the concentration as a result of which current flows. So when these 2 becomes equal, no current flows. Current is flowing because there is a difference. So that's why the cell reaction is a change in the concentration as a result of which current flows. So this is the concentration cell, and these are the reactions, how the concentration cell work. Now electropotential E is given by the EMF of the concentration cell can be calculated because we know that already EMF is equals to E cathode minus E anode. So E of cell is equals to E cathode minus E anode. So can be written as ER minus EM, that is right hand side and left hand side. So here already we have derived the Nernst equation. So in the Nernst equation, what is E is equals to? E is electrode potential, and then what is the electrode potential? E is equals to Nernst equation. With the help of the Nernst equation, we know already. So we have to put here E naught plus 0.0591 by N. N is the number of electrons involved in the reaction, and log M2. M2 here is the concentration of the solution at the cathodic side, and minus E naught minus 0.0591 by N log of M. So that is the concentration of the solution which is in which the anode is fixed. Now when we put this ER minus EA and Nernst equation, then the Nernst equation, we are going to get in the final equation, you can see that E of cell is equals to 0.0591 by N log M2 by N1. So you can see that at 298 K. You can see that E naught will not appear in the final equation as it gets cancelled. So E naught we will not get, it will get cancelled. So the concentration cell formula, you can see that here, with the help of this formula, we can calculate the E, that is the potential of the cell by using this formula, that is E of cell is equal to 0.0591 by N log of M2 by M1, where M2 is greater than M1. So with the help of this formula, you are able to calculate the potential. Then this, here we have studied about this, of course, the galvanic cell, how does the galvanic cell works, and what is a galvanic cell and how does it work, where we have understood the 2 important reactions there, that is oxidation and reduction reactions. And then we have studied about the types of the electrodes. Of course, there are so many types of the electrodes, but we have to study about the reference electrodes. Reference electrodes, again, these are the electrodes whose electrode potential values will be known to us, and are used to find out the electrode potential of other electrodes. Now also we have studied about the primary reference electrodes, secondary reference electrodes, and then we also studied the construction and the working of Kalman electrode, and then the concentration cell. So what are concentration cells, and